Okay, so our next speaker is Laurie. So we are staying in the aquatic environment, more aquatic than before. And Laurie is a senior lecturer at University of Hull, and she is working on molecular ecology of invasive species. So she will show us the potential of environmental DNA for citizen science. Thanks a lot, Laurie.
Thank you very much, Eleanor and Tim. I will just get this one. Share. Is it okay? Yep.
Yeah.
Yeah, so thank you very much for the introduction. So I'm going to tell you more about DNA-based methods more generally, because I think sometimes we get a bit hung up on environmental DNA and barcoding. So I'm going to tell you a little bit about how the broad suite of DNA-based methods could be applied for citizen science approaches. So what are DNA-based methods? A few case studies from our own group, specifically on eDNA, and then applying the technology to citizen science. And then I'm going to finish off by thinking about the prospects and challenges, of which there are numerous. So I'm only going to very briefly mention them. So what do we mean by DNA-based methods? So first of all, to make the distinction that we can sample DNA directly by collecting organisms, and the Harlequin Ladybird project has been a great example of this, where people have collected the organisms themselves and sent them into the survey, and then they've been passed on to researchers to analyse their DNA. So we can collect organisms directly, but it's also a great opportunity to collect DNA from the environment. So environmental DNA is where we collect samples from water or sediment or air or feces or something else in the environment that contains, for example, slough cells of skin or decaying matter from organisms. So we have those two distinctions. And from that information, that DNA, we can work out what the species is, but we can go way beyond that. So we can describe whole communities, we can look at population genetics and sequence genomes, we can sex individuals, we can tell what they've been eating if we've got a scat sample, or for example, and we can also look at things like host-parasite interactions. So I'm just going to start off just by saying a little bit about DNA barcoding to make it the distinction between what that is and other methods. So with DNA barcoding, we might be given a specimen like a mussel here, we'll extract its DNA and then amplify the DNA with conserved primers that amplify a specific barcoding region, which is often a chunk of the mitochondrial genome. And then that DNA is sequenced using very established methods that have been around since the 70s. And that sequence is matched against a reference database. So just in the same way that in a supermarket, when you scan your product, it's matched against a reference database of all the items that are in the stock room to get the ID. So that's DNA barcoding, and it's been around for a long time, nearly 20 years now. So with environmental DNA, so we often think about environmental DNA being from water, but it isn't necessarily. So here I am taking a water sample on Lake Windermere, and what we would normally do is take the sample back to the lab and filter it, to be honest, because that's the cheap way to do it. But for citizen science, we can use a syringe and push it through a filter and capture that DNA onto the filter, and then add a preservative so that it doesn't need to be stored in the cold. It can just be stored at room temperature. So that is quite a simple piece of kit, really, that really enables this technology to be used by citizen scientists. And then that water will contain DNA from lots of different organisms, and we extract all the DNA, and then we have two options. We can either focus in on a particular species that we might be interested in with a targeted approach that uses species-specific PCR primers, or we can use what we call a passive approach, where we detect multiple species within that DNA soup. And this is a process called metabarcoding. So I'm just going to run through the targeted approach very quickly. So there are a couple of options here. So you have your DNA sample, and it contains the DNA of the species you're interested in. You can use a very simple conventional PCR using species-specific primers that just gives you the presence or absence of the species by looking at bands on a gel. Or you can use a quantitative method that uses an additional probe. I'm not sure if you can see my mouse here, but this little star is a probe that's species specific, so it adds that extra layer of specificity. And it also counts the number of copies of DNA that are in the sample, so you have some estimate of the relative abundance of the DNA in the sample too. And then with the passive approach, we have this soup of DNA, whether it comes either from the water, or so say for example, if someone's taking a kick sample or a pitfall trap, we can make those individuals into an invertebrate soup, if you like, blend them, and we end up with a pool of DNA that's then PCR'd using conserved primers. So for example, we might use primers that are specific to vertebrates or invertebrates or a particular taxonomic group like mollusks. And then they're thrown on a high throughput sequencer, which generates about 25 million sequence reads in a single experiment. And then just like with the barcoding, those sequences are aligned using and assigned using bioinformatics to identify the species that are in that mix. So this is metabarcoding. So beyond species detection, there are actually quite a few other questions that we can address with DNA-based citizen science. So for example, we can detect new species. We can look at which pathways are important. Importantly, we can look at impact. So that's something that's been mentioned already this morning. So we can work out what the species has been eating. And from a sample, we can say, yes, this species is present, and all these other species are either present or absent, so we can look at the impact that that species is having on the community composition. So some examples from our lab. So this is Rosie, a PhD student of mine who finished a couple of years ago. And Rosie was working on macroinvertebrates, and particularly Dreissenid mussels and Dichrogamurus shrimps. And one of her chapters involved going out and sampling rivers to do kick samples. And she wanted to know whether DNA-based methods were equivalent to sampling using a kick sample. So do you get the same information from a DNA-based sample as you do from a kick sample? And during the course of this experiment, so she collected 3-minute kick samples, water samples, and also sediment samples, the latter two both for eDNA. And she metabarcoded them. And what she found was that there was a sequence in there that was Gammarus fussarum, which hasn't been recorded in the UK before. So this caused us a bit of head scratching. And Rosie looked into this in a bit more detail, and she found that these sequences were present in all 3 sample types and in 6 of the 8 catchments that she'd studied. So really quite widespread in the country, even though no one knew it was there. So obviously this needed some verification. So Rosie went back to 2 of the catchments and sampled 40 Gammarus from each of them. And she then verified the samples using morphology and also DNA barcoding, and they were confirmed as fussarum, as shown in the middle here. And then what she did, she actually went to the Natural History Museum and looked through their archives, so that they have a gazillion jars of macroinvertebrates, and she found fussarum in the archives dating back to 1964. So it's been present for a lot longer than we thought. So this is just one example of how DNA technologies can find cryptic species that we didn't know were there before. And actually about 18 months after doing this study, one of my other PhD students found another non-native amphipod for the first time. That's Crangonix floridanus, using a very similar kind of approach. So this certainly isn't a one-off example. So a big part of Rosie's project was working on Dreissena mussels, and she developed qPCR and standard PCR assays for quagga mussels, and then went and surveyed the River Wraysbury, which is the main area for quagga mussels in the UK. So they were first reported in 2014 in this big black blob on the map here. That's Wraysbury Reservoir. So she sampled from the reservoir and downstream, kick sampling again, and also comparing PCR, qPCR, and metabarcoding. And you can see from this table here, so every positive, every plus on the table is a positive detection. So out of 3 replicates, you can see for PCR and qPCR, she has 100% detection. And the numbers in the brackets here correspond to the number of individuals that were found during the kick sample. So detection rates were higher with eDNA than with kick sampling, and they're also higher with the targeted approaches than they are with metabarcoding. And we also find that there's a gradient, so a nice relationship between the distance from the source going downstream. So the DNA concentration decreases as you go further away from the reservoir. So we've done a lot of work over the last 6 years or so, mainly on metabarcoding, and we've done a lot of work on fish in lakes to develop a water framework directive classification tool, which is now being used by the UK government agencies. So I'm going to completely dismiss all that work, 6 years worth of work, and just say that, so because it's more relevant to this talk today, throughout all that data collection, we've gathered data from 110 lakes, 556 ponds, and 11 rivers. Now that's a lot of samples, a lot of data, and a lot of species records. So that's my point. So this is one example data set. So we've been doing a lot of work on Windermere as our kind of like flagship case study. And an example of the fish data sets that we're pulling out from Windermere. So we sampled on Windermere over 3 sampling time periods, and we also compared to established methods, which are gill netting, basically. And we found that we can detect 14 species of fish with eDNA compared to 5 with gill netting, that we get a good relationship between the eDNA data and the abundance data, whether it's determined from long-term rank information or biomass. That DNA is quite heterogeneously distributed in the water, which is really good because that means we can say stuff about the distribution of the actual species. And this also corresponds to trophic status. So in Windermere, there's 2 basins, and they differ quite dramatically in their trophic status. And the distribution of the fish DNA is consistent with that. So we've now scaled this up to look at 101 lakes across the UK. And I just pulled out, for the purpose of this talk, the invasive species that we found. So actually, I think maybe less than I would have expected, but we found so the white blobs on this map are the 101 lakes, and the coloured in ones correspond to where we have a positive detection of a non-native fish. And we found, so in total, 40 fish species and 41 mammals, because water bodies accumulate DNA from the surrounding landscape as well. So we can also say a lot about what's present in the local area. And we found 5 invasive fish and 4 invasive mammals, so they're shown here. So my point with this is that one sample of water contains a lot of information. It's not just one species that's in there, it can be a whole community, and not just aquatic animals, but also animals that are living in the landscape. And for a small research group like ours, trying to collect water samples on this kind of scale was impossible. So we had to rope in members of the environment agencies that we were working with. But actually, these kind of projects can massively benefit from citizen science. So my final case study is about scats. So we've been working on otters and mink over the last couple of years, collecting spraint samples or scat samples and metabarcoding them to work out what otters and mink have been eating. And you can see from these pie charts here that, so this is data that's been published, but the data collection on this is still very much ongoing. So we've got quite a small sample size for mink, but we can see that mink are focusing more on birds and mammals, whereas otters are focusing more on fish. So this is, really useful information that we can use to study the ecology of invasive species and how they're interacting with native species. And from these scats, we can also get we can work out what the sex of the individual is, and we're also doing some individual genotyping, which is possible as long as you have fresh samples. And these kind of scats can be quite easily collected by volunteers. So we've had like first-year students, for example, collecting the samples. So talking more about citizen science, so what the absolute flagship study in eDNA was actually carried out on citizen science, 2015, which was very, very early on in the days of eDNA. A study was carried out, led by the Freshwater Habitats Trust in the UK, to see whether their volunteers could collect samples that would give accurate records of great crested newts. So they managed to recruit 86 volunteers who went out to sample water from 239 ponds across the country. And they had really good detection rates. So they knew from historical records, recent historical records, whether great crested newts were expected to be there or not. So they compared the volunteers' results against the existing records, and they also found really good results when comparing the results from the volunteers against experts. So quite a simple procedure to collect these samples, and there's really good training videos available online now. So we actually have a couple of citizen science projects going ourselves, but they're in their very, very early stages. So one is it's called the 1000 Rivers Project. And this is a project that's got a lot of different partners in different countries. So I would say that our main partner is DNAbased, which are a DNA-based monitoring consultancy with their headquarters in the UK, but they really work globally. And DNAbased have designed a citizen science kit for sampling DNA, which is suitable for all ages. So, children can use it, and there's really good training videos as well. So our idea with this was to crowdfund the project, to get stakeholders who have a vested interest in their local rivers to fund sample collection. So we're asking them to donate £450 per river. And one of the main goals was to try and study the distribution of invasive pink salmon. So that's why we kind of focused the sampling in Scandinavia and Canada and in Iceland to begin with. And those samples will also be meta-barcoded, so we'll get information about the wider fish communities as well. And so far we're up to about 150 rivers, I think, although I think this bar chart's maybe slightly out of date. So quickly finishing by prospects and challenges. Very quickly going to say the technology is absolutely off the scale. We're now talking up to 15 terabytes of data in a single experiment, and we can now take this technology into the field with portable sequencers like the Oxford Nanopore MinION. And this has recently been applied to monitoring Dreissenid mussels, although I haven't seen it being used by citizen scientists yet. That maybe is a leap. And as well as the sequencing technologies, we're also getting better at the kind of material that we can sample. So you can get eDNA from air, for example, or there are projects that are either using natural or artificial salt licks to monitor things like moose. But I think the most interesting one, I think partly because I really love insects and pollinators, is that we can swab flowers for pollinator DNA to understand interactions. And there's this beautiful interaction network was published by Thompson and Sigsgaard a couple of years ago. So there's lots of developments in terms of the way that we sample. So it's not just about the water. So some pros with citizen science based methods with DNA and citizen science. So in terms of motivations, people are learning something new and getting involved in some sexy science, and maybe finding out about stuff that they had no idea was living in their local environment. It's quite straightforward to collect a sample, so children can do it. My daughter, who's 8, has had a go at it. You don't need any specialist ID or taxon knowledge. We can standardize and scale up the methods, and it's a non-invasive way of sampling. And then finally, one sample, as we've seen, contains a lot of information, not just on one single species. And finally, finishing off with the challenges. So anyone that's filtered water through a syringe will tell you that the novelty very soon wears off. And also it's a bit of a downside that you're not actually getting to see the organism. And unless you're doing the portable sequencing in the field, which is unusual these days, feedback time can take a little while because, you know, sample processing is quite involved. Everyone says that DNA-based methods are really cost effective, but you're still talking a minimum of about £50 per sample. So it's not as if it doesn't cost anything. And we need to have good training to avoid false positives and negatives. And the very last point that I want to pick up on, I think is really relevant, and I'm really pleased that I'm following Jodie here because it's really Australia leading the way, is what we do with the DNA records. So at the moment, it's not easy for citizen scientists to access DNA-based records at all. And there was a great paper, so I've been harping on about this, as Helen will know, since 2015. But there was a really nice paper published just in December about how we can make DNA-based records accessible in global databases. So at the moment, researchers are submitting their sequences to a number of different repositories. For example, the NCBI's sequence read archive, so that's a prerequisite for publication. But these databases are aimed at researchers, obviously, so they know that they're no good for non-specialist citizen scientists. So we need to make sure that we're adhering to FAIR principles. And the good news is that thanks to Darwin Core, we can now, so those platforms are ready to accept DNA records. They have been set up in that way. And Atlas of Living Australia have really pioneered this. As far as I know, it's the only country that are actually submitting eDNA records, and they are queryable. You can go onto the ALA and search for them. Some countries, so the US, for example, has its own aquatic eDNA Atlas project, but it would be really great if we could submit our records all onto GBIF and really get on board with that as a community. And I think that's very important to keep that engagement with citizen scientists. So that's me done. My key messages, I'm not going to read those out because I'm running out of time. But yeah, just to say that I hope that we can have a think about those records and different aspects going forward. And I'll just say a very big thank you to Tim and Eleanor and the members of the working group, and to you for your attention.
Thanks a lot, Laurie. Another very fascinating perspective with environmental DNA. I'm really impressed by the number of the records and data you collected. So, wow.
Thank you.
Great job also. And we have our, yeah, no, it's true. And we have our first question from the chat. So the first is, there are a couple of questions about the false positive. One is from Leaf. So how do we deal with cross-contamination between water bodies when samples are collected by the public? Because they can wear waders infested by another species, so you can detect a false positive. And also with false positive, Franz asked, there is a way to identify such false positive apart from using the traditional methods of sampling.
Yeah, so
I mean, related.
They're really important questions, and something that the field is really preoccupied with. And So I might direct you actually to one of the training videos that NatureMetrics or the Freshwater Habitats Trust have produced, because I think it may be quite reassuring to see how those samples are collected in real life. So there are lots of, you know, simple and applicable guidelines to follow. So, for example, you know, if you're sampling in a pond or sampling in a water body, you avoid standing in the water so that, you know, you're not cross-contaminating. So you you know, you wear gloves, everything's sterile, the kit is sterile. Unfortunately, all disposable plastics, which is a real problem. But in terms of reducing the cross-contamination, that's a really important thing. So I think the citizen science projects that I am aware of, and certainly the 1000 Rivers project, so we're just getting people to sample one water body at a time. So I think for those projects that are on the go at the moment, it's less of an issue, cross-contamination between projects. But actually, just thinking about it, getting that biosecurity message across is really, really important in the training. And I think that's something that I haven't seen in any training videos so far. So it's definitely something that we should think about, you know, especially when we're promoting citizen science for invasive species. There are lab steps we can take in terms of verification for false positives as well. So we do a lot of replication and use controls and collect blanks, which is really important as well. So obviously, if we had a contaminated blank, that would flag up a false positive. Does that
Thanks
answer?
a lot, Laurie. Before going to Quentin, Tim asked, how do you provide feedback to the volunteers? And okay, you told us about training videos, so you provide training video, but also training in presence to the volunteers.
Yeah, so I think that the feedback. With the 1000 Rivers project, it's quite easy to provide feedback because it's quite a small stakeholder community, and so they, you know, we're talking. I mean, in the UK so far, I think we've recruited about a dozen stakeholders. So it's that's quite straightforward. I think if you were you know, getting lots of different stakeholder groups and more people involved, then yeah, having some sort of app where you could like, you know, it's similar to Frog ID, where, you know, you could feed back and have like some sort of chat going with people would be really good. But I think that, you know, these things will be much easier if the records were in the public domain, which they're not at the moment. What was the other question? Feedback and
yeah, feedback to volunteers. And did you receive feedback also from? Do you provide feedback?
Oh, the training
to volunteers, and if you receive also feedback, yeah, and training.
Yeah. So actually with 1000 Rivers, so we have NatureMetrics produced a training video. And so I haven't actually done any in the flesh myself because of COVID. Basically, we've not been out over the last year, but NatureMetrics have. So they've also done that with the Freshwater Habitats Trust volunteers. So yeah, providing that on-site training. I think both methods are essential, really, because I think, you know, it might be that you go on the face-to-face training, but then forget something slightly. So, you know, having the videos is really useful as well. But when you receive the kits, so the kits have really straightforward instructions on what to do. It's very, you know, very clearly set out and nice pictures and that kind of thing. So, you know, at least you've got that to refer to when you're in the field.
Thank you very much. And did you receive positive feedback from the volunteers? Quentin, I'm coming, don't worry.
Yeah, so actually I don't, so this is something I don't know. So the 1000 Rivers project is at a too early stage yet really to have that kind of feedback from the volunteers. So I would like to, I'll follow that up in the future. I'd be really interested, next time I see, so it's Jeremy Biggs, who's from the Freshwater Habitats Trust. I know him quite well. Next time I see him, I'm going to ask him what the feedback from the volunteers was, because being involved in, so at the moment I'm involved in an STSM that's about motivations of citizen scientists, and I'm really, really desperate to do a big, like, you know, a questionnaire to understand what would motivate people to do DNA-based monitoring. So, so yeah, it'd be really good to know from the Freshwater Habitats Trust what their experience was. Can just say though, so they recruited, they have a project called PondNet, and this was part of that project. So they recruited volunteers that already had a vested interest in sampling ponds. And this was first carried out in 2014, and they've maintained their volunteers, and they're repeating their sampling every year. So so they must be pretty motivated, because they've stuck with the project. So to keep that ongoing for 6 years is really, really impressive.
Yeah, I agree. Thanks a lot, Quentin, for your patience. It's your time.
That's fine. I'm the only one who can't be bothered to type. That's why. Hi, Laurie. Yeah, so I tried one of the Nature Metrics kits a couple of years ago, and so it was really interesting. I just bought it for a birthday present for myself, basically.
What?
So that's the kind of person I am. So, but it was interesting. I mean, as an exercise. So some of the sort of observations I made from using it were that it's far too expensive.
Oh, it is expensive. Yeah.
And I can think of about 1 million different things I want to do with it, but I couldn't afford to do them. It was frustrating that the primers that are used were specifically for vertebrates, because in the lakes I was sampling, I know there are invasive crayfish in there, and they weren't picked up at all.
Yeah.
Well, they may have been picked up, but I never got the data. I just get a list of species. And there were also species on that list that I really didn't expect. Some were surprised, really didn't expect, but I've got no way to try and verify that.
Okay.
And there were and there were species on there. There weren't species on there that I know existed in the lakes that are vertebrates, which surprised me too. But I guess that can happen. So it speaks to the motivation, though, that I am very motivated to do it. But at the moment, because of the cost, I think the projects have to be very top down. And one of the things you see in bird watching, in botanizing and stuff like this, it's very much about people doing it for their own benefit and sharing their data. And only when eDNA becomes cheap enough that we can have our own little projects and share our data, and there's platforms to do it, will perhaps people be more motivated to do things.
Yeah, I completely agree. And I think at the moment, it's targeting particular stakeholder groups that have a vested interest in using it. So, for example, the Rivers Trust that we're working with, they're quite wealthy organisations, and they have a good membership, and they can afford it. For them, £450 to get all that information is actually, not extortionate. The technology's, it's getting cheaper, but I completely agree about Nature Metrics, because I've got a PhD student at the moment who's working with them on zebra mussels, and so we're using their filters, and we can't get the cost below £15. Even on mates rates. So it's just something stupid like a plastic filter. They are quite pricey, which is really quite frustrating. So we applied to the BBSRC, one of the UK research councils, last year for funding to take the 1000 Rivers project further. And I think there are we didn't get it, of course, otherwise I'd have been talking about that instead. But we, you know, I think that's one way of taking this further. So, you know, it's going to be large scale investment from the part of the research councils and other funding bodies to enable citizen science, rather than you know, members of the public paying for the samples themselves. But yeah, just going back to your point about the vertebrate assay. So there are loads of different assays. It doesn't have to be vertebrates, and it's harder for invertebrates. It's not impossible, but it's harder. And you know, it may be that if you're interested in a particular group of invertebrates, you would target that specific group rather than invertebrates as a whole. But yeah, I can understand. Yeah,
kind of, you want to see all those millions of hidden things, so the invertebrates are kind of interesting. Whereas fish you can catch and you can see and identify. But
yeah, definitely.
Okay, thanks a lot, Laurie. Sorry, there are 2 questions more, but